So for today, our lecture will be more about a review of the cell cycle. So for the human life cycle, it actually begins at fertilization when the egg cell inside the woman and the sperm cell from a man fuse to form what you call a zygote. But before the formation of zygote, during the formation of the gametes from the female and the male, the number of the chromosomes is reduced by half and returned to the full amount when the two gametes fuse during fertilization. Hence, the chromosome number that they have, or what you call the ploidy, the term referring to the number of sets of chromosomes for the sperm and the egg before fusion, is actually in haploid stage. Okay, when you say haploid stage, these are cells that have only one set of the chromosomes, and they are usually abbreviated with letter N. And then once there's already a successful fusion of the sperm and an egg, so that would be the time that the zygote, or the fertilized egg, will already have what you call a diploid stage, or um, the combination of two sets of chromosomes coming from the maternal and the paternal parent. Okay, so once the zygote is already fertilized, of course, it will undergo the necessary changes and development, like the gastrulation, the blastulation, the neurulation, and eventually will reach the the level of development that includes now the different cell differentiation for the formation of the different types of cell in the body, and of course for the development of the other body parts of the body, like under organogenesis. Okay, so once there's already a juvenile, okay, it will also undergo further development until an individual becomes an adult as well, who's already capable of reproducing and is already, or do already have, the mature type of cell. So again, let's review the different types of cell of the body. First, we have the somatic cells, which pertains to the cell composing of the bulk of the body, such as the nerve cells, muscle cells, and the kidney cells. So these somatic cells are all of the other types of the cells in the body except your gametic cells. So for the gametic cells, it pertains to the reproductive cells, such as the ova of the female and the spermatocytes of the male. Okay? So multicellular organisms use actually mitosis for growth, cell repair, and cell replacements. So for mitosis, usually it is being what you actually develop are actually the somatic cells. Okay. So for the cell cycle, it is a series of events that takes place in a cell as it grows and divides. So a cell spends most of its time in what we call the interphase, and during this time it grows, replicates its chromosomes, and prepares for cell division. Then the cell then leaves interphase and undergoes mitosis and completes its division. So with the schematic diagram that you're seeing on your screen right now, as you see, the interphase actually has subdivisions, like the G1 phase, the S phase, the G2 phases, and it shows you what type of development happens. So let me further discuss it to you. So the cells go through two basic phases. So the first is actually the interphase, and then the rest would be under mitosis. So in the interphase, we have the G0 phase, wherein there's still normal cell functions. For the G1 phase, or the growth 1 phase, this already prepares you for the cell for cell division. So cell continually imbibes water and nutrients and it grows. So next would be the S phase, or the synthesis phase. This is already when the DNA replication and chromosome duplication happens. And the G2 phase, or the growth 2 phase, is where the RNA and protein synthesis happens, and the centriole synthesis and other raw materials needed for cell division are being produced. Okay? So let's differentiate mitosis and meiosis. So again, cell division that occurs by mitosis produces new cells for growth and tissue repair, while under meiosis, it actually produces your gametes or the sex cells. Again, it's the sperm cells in males and oocytes or egg cells in females. So for mitosis, it is actually a nuclear division that has the inclusion also of what you call the cytokinesis. Okay, so it produces two identical daughter cells during prophase, pro-metaphase, metaphase, anaphase, and the telophase. So interphase is often included in the discussion of mitosis, but interphase is technically not part of mitosis, but rather encompasses the stages G1, S, and G2 of the cell cycle. So for the interphase, it is the time between the cell divisions, and this is where DNA is found as thin threads of chromatin in the nucleus, and DNA replication occurs during the interphase as well. So these are already the important events that you should know when it comes to understanding mitosis. Okay, so um, in interphase also, organelles other than the nucleus duplicate during the interphase. So if you check on the image here, okay, you see that this is the nucleus, and then the chromatin, okay, of the DNA is being replicated here. Okay, so and then eventually when it enters the mitotic division of the cell cycle, the first phase would be the prophase. So in the prophase, the chromatin condenses into chromosomes. So again, in the interphase, you call it chromatin, and then it condenses to chromosomes once it enters the prophase stage. So the centrioles move to the opposite ends of the cell, and the nucleolus and the nuclear envelope disappears. And then microtubules form the near centrioles and project in all directions. And then the spindle fibers project toward an invisible line called the equator and overlap with the fibers from the opposite centrioles. So here you see the spindle fibers, these are the astral fibers, and then the centrioles. This is the so-called equator. And then here you see that the chromatids are condensed into what you call now your chromosomes. Okay. Next would be metaphase. In metaphase, the chromosomes align in the center of the cell in association with the spindle fibers, and some spindle fibers are attached to the kinetochores in the centromere of each chromosome. So again, this is the spindle fiber. The kinetochore is the one in the middle, and then the chromosomes. So as you see, there's already a pattern forming because of their alignment in the center. So that depicts now your metaphase stage. Next, for anaphase, this is when the chromatids separate, and each chromatid is then referred to now as chromosomes. So once the chromatid separates, eventually it's now called an identical chromosome. So thus the chromosome number is double, and there are two identical sets of the chromosomes already. So the chromosomes are assisted by the spindle fibers. They move toward the centrioles at each other of the cell, and separation of the chromatid signals the beginning of anaphase, and by the time anaphase has ended, the chromosomes have reached the poles. So in here, other than the separation of the chromosomes, you can already see also a part here that the cleavage furrow is already starting to be seen or to be observed. Okay. So next is the telophase. This is when migration of each set of the chromosome is already completed, and the chromosomes unravel to become less distinct chromatin threads. So it just appears that they are trying to go back to the original look that they had before. Okay, so the nuclear envelope forms again from the endoplasmic reticulum, and then the nuclei form, and cytokinesis or the cytoplasmic division continues to form two cells. Okay, so what's already or what you can find on the right is also present on the left side before it fully separates from each other. Okay, so again, for telophase, what's more important for you to know is that there's already the appearance again of the nuclear envelope. And then mitosis is actually complete and a new interface begins once they have already been completely separated, or the daughter cells has already been completely separated from each other. So the chromosomes have unraveled to become chromatins again, and then cell division has produced two daughter cells, each with DNA that is identical to the DNA of the parent cell. Okay. So this is actually the best example or model for you to observe mitosis. This is the onion root tip. So it is more likely used um, to observe um, mitosis because it's readily available. It's very easy to grow. Just even if you're just at home, you can actually grow onion root tips overnight. Okay, so again, for cell division, it's mitosis or the nuclear division plus the cytokinesis or the cytoplasmic division. So in here, image number 1 is actually the interphase. Image number 2 is the prophase stage. 3 is the metaphase. 4 is anaphase. 5 is telophase, and then 6 is cytokinesis. So that's how it actually looks like under a microscope with the use of an onion root tip. So let's go now with meiosis. So meiosis is or do actually comprise of two successive nuclear divisions that occur. So the first one is meiosis I or the reduction phase, and meiosis II is the division phase. Okay, so meiosis produces 4 haploid cells. So if mitosis produces 2, what happens now with meiosis is that you can produce 4 haploid cells. Okay? So the old name for meiosis was reduction or division. And then meiosis 1 reduces the ploidy level from 2n or diploid to haploid. So that's reduction. While meiosis 2 divides the remaining set of chromosomes in mitosis-like process you call division. So most of the differences between the processes occur during meiosis 1. So it's just like doing mitosis twice. Okay? But then again, the composition of the cells being produced um, in meiosis are sex cells and not somatic cells. So the differences of mitosis and meiosis, of course, for the cell type, for mitosis, it's what you call the somatic or body cells. For meiosis, it's the gametic or sex cells. So are there genetic variations? Of course, for mitosis, there is none. So because you just copy the maternal or the genetic copy of the parent. In meiosis, there is. It's because of the exchange of genes during synapses or crossing over of the genes from maternal and paternal um, no, chromosomes. Okay. So for the product, for mitosis, you have 2 diploid daughter cells, while for meiosis, you will be having 4 haploid daughter cells. And then for the stages of mitosis, it's a single cycle, part of the cell cycle that includes your interphase and then your mitosis stage proper. And then for meiosis, we have terminals with 2 stages, We have Meiosis I and then Meiosis II. So crossing over during meiosis, this actually what's happening during the fusion of the maternal and the paternal genes. Okay, so we called it we actually call it as crossing over or the recombination stage. So the homologous pair moves close together, and then the chromatids may exchange their genes. So once that happens, you can already depict with the use of different colors here. If you have, let's say for paternal, let's use the blue one and then red one for the maternal. So once crossing over or recombination already happens, so it can be like this, wherein the maternal and the paternal is already intertwined with each other. So depending also on how much, okay, that crossing over do happen. Okay, and then for the first cell division, homologous pair of the replicated chromosomes also separate. So from this one, it also separates, and then eventually sister chromatids also separate. So giving you 4 haploid daughter cells. So by comparing, you can see that there's already variation from the from the 4 haploid daughter cells. Okay, it can be a pure paternal, two can be a mix of the maternal and the paternal, and then another one for the maternal. Okay, so that's how actually um, that actually tells you what variations are. Okay, so for example, in a family of yours who can be a child who inherits characteristics from their father, o kaya naman if a child um, that can get the characteristics of the mother, meron naman na pareho na meron silang characteristics of both the mother and the father. Okay? So for the stages in meiosis I, so again, meiosis I is actually known as the reductional division, and prophase I is the longest phase in this stage of meiosis. So here you will see different images and stages of meiosis I. So the first one would be the leptothene with thin thread-like chromatins. Here, this one. You see that there's a centriole, the chromosomes, a centromere. This is the leptothene phase. And then you have the zygotine. This is where the occurrence of synapses or pairing of homologous chromosomes happen and start of the formation of chiasmata or crossing over. So this is the zygotine stage. And then the pachytene with pacus, thickened stumps of chromosomes. So this one. And then the diplothene, this is the occurrence of the terminalization or the separation of chromosome legs. Chiasmata proceeds to the terminal ends. This one. And then the diakinesis, or this is when the chromosomes at maximum contraction already. Okay, this one, diakinesis. So the metaphase 1 happens with the alignment of the bivalents of the equatorial plane. Anaphase 1, separation of the bivalents to form 2 univalents. And then the telophase 1 is the formation of 2 haploid daughter cells. So from there, it would eventually continue to meiosis II, which is already the equational division. So this has, again, like the stages similar to mitosis. So prophase 2, univalents contract. Metaphase 2, alignment of univalents at the equatorial plane. Anaphase 2, separation of the univalents into 2 sister chromatids and migration to the opposite poles. And then telophase 2, which is already the formation of 4 haploid daughter cells. So for the morphological changes during maturation of gametes in the spermatogenesis, so again, spermatogenesis is actually the maturation of the sperms. And for males, the maturation of their sperm actually happens or begins at puberty. Okay, so later on I'll be discussing to you more of what is the difference of the maturation of the egg as well. Okay, so at birth, male germlators can be recognized in the sex cords as large pale cells surrounded by supporting cells, and shortly before puberty, sex cords acquire lumen and become seminiferous tubules. So if spermatogenesis is the maturation of the sperms beginning at puberty, we also have what you call spermiogenesis, or the maturation of the sperms, okay, or the sperm cells. So in this one, in this image, in the longitudinal section of disease of abital, the Blapsmerk saga, you will see here the different stages of spermatogenesis, correlating the different divisions in meiosis. So in there, you will see leptotene, pacotene, zygotene, and then some of the cells also do undergo already the second meiosis, or meiosis II, with the presence of the anaphase, the telophase stages. Okay? So coming from the longitudinal section of AB, let's look into now the longitudinal section of A mammalian testes. So in here, you will see the different stages of spermatogenesis as well for higher animals, including humans. So you will see here that you have the retitestes, the lubulus testis, and then the tunica albuginea, which actually surrounds um, the testes. And then you see here the loop of seminiferous tubules, and then the epididymis. So the maturation of the sperm is actually in the seminiferous tubules. Okay, so once you check in the seminiferous tubules, you will see here the different levels of development of the sperm cell. So the seminiferous tubules, you have here the peritubular tissue that covers it. So spermatogonium is more likely adherent to the seminiferous tubule wall. And then you have this primary spermatocyte, the immature spermatids, and then the mature spermatids there in the middle. Okay, so the younger cells are more adherent to the cell wall, and then, or to the lining of the seminiferous tubules, and then going to the middle is already the mature spermatids. Okay. So seminiferous epithelial cycle. So layers of germ cells all develop in a coordinated repeating program known as seminiferous epithelial cycle, or what you call the SEC. So in rodents, successive stages are arranged in cylindrical segments along the seminiferous tubules. So under a microscope, the testis tissue looks like this. Okay, in a cross section, the testis is like this, but for a transverse section of the seminiferous tubules, as what I have explained earlier, you see here that you have the basement membrane. The younger types of cells, spermatogonia, are more adherent to the basement membrane. So it undergoes growth and mitosis to be able to become a first degree spermatocyte, and then undergoes meiosis I to be a secondary or second degree spermatocyte, and then eventually become a spermatid after meiosis II. Okay? So Sertoli cell is actually a type of cell that produces food for the to support the growth of your spermatids. So for the cycle and wave, its existence has been demonstrated in many vertebrate species. They are features of vertebrate spermatogenic organization that reflect the need for rigorous sperm production. So the cycle of the seminiferous epithelium is synchronous mitotic meiotic divisions. There are 6 stages in man and happens in 4.6 cycles from sperm release, or 16 days per cycle. So the waves in the seminiferous epithelium in rodents and in other animals, they do have these waves, but no waves in human seminiferous tubules. So for the cycle and the wave, the designated stages of the seminiferous epithelial cycle in rat is denoted by the Roman numerals 1 to 14. So as you see here, if this is a section of the seminiferous tubules, some of them are numbered, right? Okay, so that depicts the stages of sec in a rat. So 12 of such stages, like 1 to 12, have been described in the mouse. So up to here. So this is one wave, as you see. So stage duration is precisely timed, and the complete spermatogenic cycle was determined to be about 8.6 days in the mouse and 12.6 days in rats. So each section of the seminiferous tubule corresponds to the highest developmental stage of the sperm. So as you see, for of course for the peaks, okay, so those are the denoted stages wherein there's the highest developmental stage of the sperm. So but just by looking at the image or the figure, you will know that the highest peaks are during stage 9, 10, and 11. Okay, so the least stages are around 5, 6, and 7. Okay. So in this one, or in this table, it will show you the characteristic cell associations at each stage of the cycle of seminiferous epithelium in the mouse testis. So you have your A for spermatogonium, INT for intermediate spermatogonium, etc. So this is actually what happens or what is being produced during each stage of the cycle, and um, what type, okay, are being produced. Okay. So morphologies of the spermatogenic cells. So see here you have different types. You have the dark types. Okay. These are just you know images that actually would explain to you how they look like um during their development. So next, we have the spermatogenesis 1 for the spermatogonial phase. So type A spermatogonia, the stem cells of the seminiferous tubules are usually dark in color. And then for intermediate spermatogonia, it is committed to the differentiation. It's more likely pale in color if you um, tend to observe it under the microscope. And then type B spermatogonia, it's differentiated from type A spermatogonia, the last event in the spermatogonial phase. Okay. And then spermatogenesis 2, or the spermatocyte phase or meiosis phase. So primary spermatocyte is abbreviated as for haploid, or it's in a haploid stage. So it's produced by type B spermatogonia. First meiotic division, homologous chromosomes are crossing over and give rise to secondary spermatocyte. While for the secondary spermatocyte, which is already at um, 2N, it happens or it's being produced during the second meiotic division and give rise to a spermatid. So it becomes diploid already. So this picture that you see on your screen right now is a schematic diagram of spermatogenesis in the pre-puberal and adult mouse testes, showing the relative volumes and characteristic morphology of the respective cell types. Okay. So spermiogenesis is the spermatid phase. So it has different phases as well. You have the Golgi phase. This is where the proacrosomal granules, acrosomal vesicle appears, and axonemal complex. So you already have here the sperm tail. And then you have the cap phase, or the formation of the acrosomal cap. And then the acrosome phase, or the manchette, this is already the flagellum development. And it already, or the the nucleus of found in the acrosome of the or in the head of the sperm is being condensed. And then for the maturation phase, residual body is pinched off and phagocytized by the Sertoli cells. So now let's go to oogenesis. So if we have spermatogenesis, we also have the oogenesis, which is the maturation of oocyte that begins before birth. So primordial germ cells that arrive in the gonad of female embryo will differentiate into oogonia, and then from oogonia it will undergo mitosis and then meiosis and then prophase 1 and then so on and so forth. So if again the spermatogenesis, or the maturation of the sperm, happens during the puberty period um, of males, for females, we already have the oocyte even before birth. Okay, but then again, it just activates, or it just undergoes um, development if we also reach puberty. But originally, we already have oocytes, or for females, they already have the oocytes beginning even before birth. Okay, so it is one of the earliest cells that are being produced in the body compared to the males. So the development of the egg cells are just arrested for a very long time until a female is already able to undergo further development and maturation of the egg cell. Okay, so for the maturation of oocyte, it actually again continues after puberty. So near the time of birth, all primary oocytes enter the protein stage and do not finish meiosis I until puberty, because of the oocyte maturation inhibitor. Okay, so this is the reason why um, the cells are arrested and they don't continue to mature until puberty is reached. Okay, so at puberty, a pool of growing follicles is established, and there are 15 to 20 follicles that are selected from the pool and differentiate into three stages. So the three stages of growing follicle is what you call the primary or preantral, the secondary or antral, also called the vesicular or the Graafian follicle, and then we have the pre-ovulatory follicle. So for oogenesis, so in here you will see how the differentiation of primordial germ cells into oogonia begins shortly after their arrival in the ovary. So by the third month of the development, some oogonia give rise to primary oocytes and enter prophase of the first meiotic division. So this prophase may last around 40 or more years and finishes only when the cell begins its final maturation. So during the period, it carries around 46 double-structured chromosomes. So this is the primordial germ cell. After the first meiotic division, you will have the oogonium. After the second one would be the primary oocyte in prophase. So in here, this is actually the segment of the ovary at different stages of development. So letter A here is at 4 months. The oogonia are grouped in clusters in the cortical part of the ovary. And somehow mitosis other have differentiated into primary oocytes and entered prophase of the first meiotic division. Okay, so if this is the oogonia, these are already the primary oocytes at first mitotic division. And then here on the 7th month, this is almost all oogonia are transformed into primary oocytes in prophase of the first meiotic division. And then lastly, for a newborn, there are no oogonia. Each primary oocyte is surrounded by a single layer of follicular cells, forming the primordial follicle. So oocytes in this stage have entered the diplotin stage of the prophase, in which they remain until just before ovulation. So only then do they enter metaphase of the first meiotic division, when a female is already viable to do it. So in here, you see the primordial follicle consisting of primary oocytes surrounded by a layer of flattened epithelial cells. So these flat epithelial or follicular cells are surrounding the primary oocyte. So this is the nucleus. And then in here, for the growing follicle, this is the early primary or pre-antral stage. This is how it looks like. So as the follicle grows, follicular cells become cuboidal. So from flat here, from having flat cells, in growing follicles, they do become cuboid in shape. Okay, and begin to secrete the zona pellucida, which is visible in irregular patches on the surface of the oocyte. So this is already the beginning of the zona pellucida. And then mature primary or preantral follicle looks like this with follicular cells forming a stratified layer. So when you say stratified layer, um, it's stuck with each other. So from a single line of cells, it becomes stuck. And you see here now that most of these cells are actually granulosa cells that surrounds the oocyte and the presence of a well-defined zona pellucida inside. So the secondary antral stage follicle is like this. The oocyte surrounded by the zona pellucida. This one. So it's off center. The antrum has developed by fluid accumulation between intracellular spaces, and note that the arrangement of the cells of the theca interna and the theca externa. So in here, this is the antrum. This is more likely where the fluid is. And then you now develop the theca interna and then the theca externa. So these cells that are more adherent to the primary oocyte is actually what you call the cumulus oophorus. So for the maturation of the oocyte, so again, primary oocyte in division, showing the spindle of the first meiotic division, and then secondary oocyte and first polar body. So you will see here also a polar body. The nuclear membrane is absent. And then for the secondary oocyte, this is the secondary oocyte showing the spindle of the second meiotic division, and the first polar body is also dividing. So for the clinical correlations for the sperm and egg abnormalities, so there are important cases wherein, of course, there is or there will be problems when it comes to the development of the egg and the egg and the sperm. So important stages in transformation of the human spermatid into spermatozoon. So usually, as you see here, this is how the sperm develops. Okay, so you have the centriole, the Golgi material, so, and then you also have the acrosomic granules, and then eventually acrosome development happens, and then here you will already see the ring structure of the middle piece. There's already a defined nucleus covered by the acrosome, and then eventually the different components of the cell condenses, and then what's left is just the head, the middle piece, and the tail of the sperm. So in here, abnormal germ cells. So A is actually a primordial follicle with two oocytes. So usually we only have one. And then B is a trinucleated oocyte. And then for C, these are various types of abnormal spermatozoa. So as you see, there can be sperms which have two heads. There can be some sperm that have two tails. Some are short, some do have big acrosomes or heads, and some are also very small. Okay. So for numerical abnormalities, so usually these types of abnormalities are more likely seen when you observe DNA. Okay. So you see that there can be translocation of the long arms of the chromosomes. So actually guys, with the coloring or the labels of this DNA, it actually pertains to a specific protein wherein it's also responsible for a specific phenotypic or genotypic characteristic of an organism. Okay, so if there is an abnormality of that certain protein from your DNA, of course, it would alter the result or the characteristic that is intended for. Okay, so when you say translocation, it can be that this part, okay, or this specific protein did not land on the expected part of the DNA and has been transferred to a different one, or it can also be uh, a genetic variation or manipulation that has been done on the DNA. Okay, so usually we have this what you call karyotyping. Okay, from here you will be able to see how the genes look like depending on the abnormality that we're trying to see. So usually there are 22 pairs of chromosomes being compared. That's for maternal and paternal chromosomes. And of course, X or Y would actually determine the gender of the organism. Okay, so one example of the clinical of numerical abnormalities would be trisomy 21 or the Down syndrome. So it is usually caused by an extra copy of chromosome 21 or trisomy 21. So as you see here on the karyotype, usually you only have two chromosomes, but here what you see are three chromosomes, specifically in chromosome number 21. Okay. So, children with Down syndrome is characterized with flat, broad face, oblique palpebral fissures, epicanthus, and furrowed lower lip. So, another characteristic of Down syndrome is broad hands with single transverse or simian crease. And many children with Down syndrome are actually mentally retarded and have congenital heart abnormalities. Okay. So next, we have trisomy 18. So for trisomy 18, you see there that there's a prominent occiput, cleft lip, micrognathia, low set ears, and one or more flexed fingers. So this one at the bottom has trisomy 13. So also they have cleft lip and palate, the sloping forehead and microphthalmia. And syndrome is commonly accompanied by polydactyly or multiple fingers. Okay. So next one is the Klinefelter syndrome. So in here, one in every 2,000 live births have male sex organs but are sterile. So they have also the feminine characteristics like some do have breast development and lack of facial hair. They are tall but they also have normal intelligence. So it's an XXY male. Okay. So they have an extra chromosome from the maternal. So next is the Turner syndrome. So characterization of this one would be having epicanthal folds, low posterior hairline, cubitus valgus, broad chest with widely spaced nipples, cardiovascular anomalies, hyperconvex fingers and nails, pigmented nevi, and sex chromatin is negative. So other characteristics are already shown in this picture. So with Turner syndrome, you see here, it patients with this one has a loss of the second chromosome. So there are also, other than numerical abnormalities, there are also structural abnormalities like the Angelman syndrome. So the Angelman syndrome is actually um, a result of a microdeletion on maternal chromosome 15. If the defect is inherited on the paternal chromosome, the opposite of it would be called a Prader-Willi syndrome. Okay? So microdeletion occurs on the long arm of the chromosome 15 in maternal chromosome, and it is characterized by being mentally retarded. You cannot speak, you exhibit poor motor development, and are prone to unprovoked and prolonged periods of laughter. So, opposite of the Angelman syndrome would be the Prader-Willi syndrome. This is also a microdeletion that occurs in the long arm of the chromosome 15, but then again, it's from the paternal chromosome. So, affected individuals are characterized by hypotonia, obesity, mental retardation, hypogonadism, and cryptorchidism. Okay, so that actually ends our lecture for today for the review of cell cycle. So if you have any questions, just let us know so that we could address your concerns. Okay, thank you.